Contrasts in human and mouse synapses have suggestions for investigation into neurological issues. Discoveries. A UCLA Drove study contrasting synapses known as astrocytes in people and mice found that mouse astrocytes are stronger to oxidative pressure, a harming unevenness that is a component behind numerous neurological problems. An absence of oxygen triggers subatomic fix systems in these mouse astrocytes yet not in human astrocytes. Conversely, aggravation actuates resistant reaction qualities in human astrocytes yet not mouse astrocytes. Foundation. Albeit the mouse is a universal lab model utilized in research for neurological infections, results from concentrates in mice are not generally relevant to people. Indeed, over 90% of medication up-and-comers that show preclinical guarantee for neurological issues at last bomb when tried in people, to some extent because of a shortage of information about the distinctions in astrocytes and other synapses between the two species. More established grown-ups' apparent intellectual decay might be identified with changes in mind-useful network. Mouse-adjusted SARS-CoV-2 and simian adenovirus vector advanced COVID-19 antibody research. Novel communications between proteins assume a basic part in the cerebrum injury recuperation measure. Astrocytes are pivotal to the turn of events and capacity of the mind, and they assume a considerable part in neurological issues that, regardless, isn't completely perceived. Injury or contamination makes astrocytes go from a resting to responsive state where they can help in fixing the mind, however can likewise increment adverse irritation. Technique. The researchers examined creating cells sanitized from mouse and human cerebrum tissue, just as cells filled in without serum societies from astrocytes chose utilizing an immunizer-based technique created by the examination's relating creator. This procedure was essential in light of the fact that the traditional strategy for choosing astrocytes by developing them in serum, a combination of proteins, chemicals, fats and minerals, tosses them into a responsive state like that brought about by contamination or injury. With the analyst system, they had the option to look at the astrocytes in a solid state and in controlled states of oxidative pressure, absence of oxygen and unnecessary aggravation. Effect. The discoveries have suggestions for fundamental and translational investigation into neurological issues like Alzheimer's sickness, Parkinson's illness and amyotrophic sidelong sclerosis, conditions whose hidden components incorporate oxidative pressure, absence of oxygen and exorbitant irritation. Since mouse astrocytes confront oxidative pressure better, the creators recommend that research center models for neurodegeneration could be designed to reduce that obstruction, delivering them more human-like. What's more, the mouse astrocytes Office for Fix because of absence of oxygen may propose another road of stroke research. What's more, neuroscientists can adopt a more educated strategy to preclinical investigations by representing contrasts because of aggravation among mouse and human astrocytes, just as metabolic contrasts distinguished in the examination.